Let me show you one example of fluorescent molecules from our own research, which is called KP1. KP1 is useful in the stem cell research. I'm sure you are familiar with human ES cells, embryonic stem cells. ES cells can differentiate into any type of cells, and ES cells can proliferate as well. The Professor Yamanaka in our institute developed a way to make induced pluripotent stem cells, iPS cells. iPS cells are essentially equivalent to ES cells. iPS cell can differentiate into any type of cells, and they proliferate. The Professor Yamanaka got a Nobel Prize in medicine or physiology some time ago. Then people are using iPS cells or ES cells for stem cell therapy. In the stem cell therapy, human ES cells or iPS cells need to get differentiated into particular cell types. Then after differentiation, they need to be injected or transplanted into the patients or animals. The problem that we have right now is the tumor formation because of the leftover stem cells. If we come up with the fluorescent molecule that can label only undifferentiated stem cells, we might be able to detect them, then we might be able to remove them for safer transplantation. So we collaborated with Professor Yontae Chan in Singapore. Professor Yontae Chan synthesizes the chemical library of fluorescent molecules. Then we also had fluorescent molecules. We exchanged a chemical library of fluorescent molecules. Then we used those fluorescent molecules for screening. So chemical library of fluorescent molecules was screened. Then we found one highly selective molecule we named Kyoto Probe 1, or KP1 in short. This is a chemical structure. The KP1 can stain, can label undifferentiated stem cells, iPS cells and ES cells. Then let me show you the fluorescent property of KP1. Would you turn off the light? Let me turn off this. Okay. This is a UV lamp, right? Then I'll show you the KP1 tube right here. So it's really fluorescent, like that. It absorbs the light, UV light. It emits the visible light. So that's why it is fluorescent, like this. Okay. So KP1 can label iPS cell colony like this, but not feeder cells. Feeder cells are like carpet for stem cells. The Professor Yamanaka's laboratory did this experiment. If you are not careful enough, iPS cell can get differentiated. Then the central part of the colony can get differentiated. Then you can get donut-shaped colony. Inside the colony is differentiated. Outside is still undifferentiated. With KP1, you can see the staining of undifferentiated part of the colony in a selective way. Then here's a human ES cell staining. Professor Nakatsuji's laboratory did this experiment. Here's the human ES cell colony. Then KP1 can stain human ES cells. Then upon the partial differentiation, just undifferentiated part of the colony was stained, but differentiated part of the colony was not stained. Then how does this work? Then we were able to understand how KP1 works. This is a summary. Professor Ueda's laboratory in our institute found that human iPS cells or ES cell have distinct expression pattern of ABC transporters. ABC transporters are like pumps for cells. If cells get something inside, ABC transporters try to get rid of it, get rid of those unnecessary molecules. It's just like a pump. But our cells have 48 different kinds of ABC transporters, but inside the iPS cell or ES cells, ABCC1 is expressed. Here's a pump, but upon differentiation, two different kinds of the pumps, ABCB1 and ABCG2, are expressed. The KP1 happens to be exported by these two transporters. That's why before differentiation, KP1 can accumulate inside the ES cells, but upon differentiation, KP1 can be removed by these two newly expressed transporters. That's how it works. I have a video here, but you can see the video in the course material where you can see donut-shaped colony. Again, inside the colony is differentiated. Outside is undifferentiated. Since the ABC transporters are involved, KP1 staining is time dependent. You can see the video in there. So this is the example of, this is one thing that you can do with the fluorescent molecules. This is one of the chemical genetic studies using the fluorescent probes.